Imagine a patient presenting with spastic paraparesis. A physical exam reveals dead, completely absent ankle reflexes, yet simultaneously their knee reflexes are hyperactive and spastic. Look at this medical diagnostic layout. On the left, the patient's routine lab printout shows their total serum B12 is completely normal. Yet on the right, their T2-weighted spinal cord MRI reveals pathognomonic symmetrical hyperintensities, glowing white lesions in the posterior and lateral columns. Their spinal cord is actively disintegrating. They are suffering from subacute combined degeneration, a catastrophic disease driven by Cobalamin or vitamin B12 deficiency. Yet right now, as this patient loses the ability to walk, their own microbiome is synthesizing massive amounts of active B12 inside the colon. The highly specific cellular doors required to pull that B12 into the blood are located strictly upstream inside the small intestine. The internal bacterial factory is entirely useless to the host, and the nutrient is simply flushed away as waste. We harbor an immense internal supply of a life-saving molecule, yet remain isolated from it by a few centimeters of intestinal anatomy. We are left entirely dependent on a fragile dietary extraction process and deeply flawed laboratory testing to keep our nervous systems intact. Cobalamin is the largest, most structurally complex non-polymeric vitamin in human biology, an intricate, asymmetrical coordination compound. Most massive biological molecules, like DNA or proteins, are repeating polymer chains, but B12 is unique. At its center sits a corin macrocycle, coordinating a single trivalent cobalt atom perfectly at its core. This cobalt forms an exceptionally weak covalent bond with carbon. This weakness allows homolytic cleavage. The bond breaks and reattaches effortlessly at body temperature, requiring no external ATP. This allows the molecule to operate like an organometallic crane. It picks up and drops carbon atoms exactly where the cell needs them to repair DNA and build tissue. Constructing this specific corin ring requires a metabolically exhausting assembly line of over 30 separate enzymatic steps. That genetic blueprint is strictly confined to ancient anaerobic bacteria and is entirely absent from the genomes of all higher plants and animals. By losing the ability to synthesize this molecule internally, mammalian evolution tethered our genomic stability and structural nerve integrity entirely to the microscopic rules of the food web. Because we cannot make it, we must harvest it. Look at the schematic of the human digestive tract. Extracting B12 is a terrifyingly fragile multi-organ gauntlet. It requires exact timing and pH shifts to succeed. In the stomach, hydrochloric acid and the enzyme pepsin strip the B12 out of the meat matrix. But that exact same acid will instantly destroy the naked vitamin. To survive, your salivary glands simultaneously drop a specialized protective protein shield called haptocorin, wrapping the B12 to save it from the acid bath. The complex then moves into the duodenum, where the environment shifts from acidic to alkaline. The pancreas releases enzymes to strip that haptocorin shield off, allowing the stomach to send down a second chaperone called intrinsic factor. This new intrinsic factor B12 complex travels all the way down the intestine until it hits the distal ileum. There, the complex binds to QBAM receptors, highly specific doorways that recognize only this pairing, to finally enter the bloodstream. If a patient develops atrophic gastritis, pancreatic insufficiency, or an autoimmune disease that attacks intrinsic factor, this multi-organ chain breaks. Absorption drops to zero. Because that daily cascade is so unreliable, the body maintains a backup plan. The hepatic vault inside the liver actively stockpiles 50 to 60% of your total B12 content. A strict, unsupplemented vegan relies entirely on this vault. Because the reservoir is so large, they feel healthy for 3 to 5 years before hitting zero and experiencing a sudden, severe metabolic crash. When that vault runs dry, 2 cellular engines break down simultaneously. The first is the cytosolic methionine synthase engine, the DNA engine. Here, B12 serves as the obligate intermediate that releases trapped folates to fuel DNA synthesis. Without B12, folates become permanently trapped in a useless state. DNA synthesis inside the bone marrow stalls, and the body begins spitting out huge, half-finished, dysfunctional red blood cells, macrocytic anemia. The second system to fail is the mitochondrial mutase engine, the nerve insulation engine. This operates deep inside the mitochondria to clear a toxic metabolic byproduct called methylmalonic acid, or MMA. When B12 disappears, the mutase engine stalls, and toxic MMA builds up massively. The body attempts to utilize the waste, synthesizing abnormal branch-chain fatty acids. These faulty fats are incorporated directly into the lipid layers of your nerve insulation, physically destabilizing the myelin sheets. Running out of B12 is the simultaneous shutdown of rapid blood cell replication and the physical decay of the nervous system's structural wiring. The extreme chemical reactivity that drives those cellular engines also makes B12 highly valuable in emergency medicine. It is used in the ICU as a literal antidote for deadly poisoning. When a victim of a structural house fire is rushed to the ER, burning plastics release lethal cyanide gas. That cyanide suffocates the patient's cellular mitochondria, halting respiration. Doctors execute the cyanokit protocol, injecting an astronomical 5-gram intravenous dose of hydroxycobalamin directly into the patient's bloodstream. The highly exposed cobalt core acts as a massive chemical magnet. It rips the toxic cyanide right off the dying mitochondria, binding it tightly to the cobalt and instantly turning the poison into harmless synthetic B12. This life-saving property contrasts with a hidden, severe danger found in routine surgical settings, nitrous oxide, or laughing gas. Inhaled nitrous oxide interacts directly with the B12 in the blood. It permanently oxidizes that central cobalt atom, flipping it from a highly active state into a dead, useless state, instantly neutralizing whatever tiny fraction of active B12 a borderline patient had left. The exact chemical reactivity that allows B12 to rescue a patient from circulatory collapse makes it exquisitely vulnerable to anesthetics. A single exposure can plunge an undiagnosed patient into spinal cord deterioration overnight. This brings us back to the opening paradox. If the engine stalls so violently, why does a standard total serum cobalamin blood test fail to catch the deficiency? Up to 80% of the B12 floating in the bloodstream is bound to the salivary shield, haptocorin. This specific form is an inactive transport reservoir that your tissues cannot actually use. In patients with autoimmune pernicious anemia, an even more dangerous testing trap occurs: assay crosstalk. In pernicious anemia, the immune system creates autoantibodies that destroy intrinsic factor. Inside an automated lab machine, these large human autoantibodies physically swarm and block the docking sites of the machine's testing reagents. The automated machine gets confused by this physical interference. It mistakenly registers the blocked chemicals as a massive presence of healthy B12. Relying solely on a total mass screening is a catastrophic diagnostic trap. To find the truth, clinicians must measure the vitamin's actual metabolic function. The gold standard for confirming this is liquid chromatography tandem mass spectrometry. Instead of looking for B12 itself, the mass spectrometer looks for the metabolic waste it is supposed to clear out. If plasma methylmalonic acid crosses the threshold of 0.4 micromoles per liter, the mutase engine has definitively stalled, regardless of what the standard blood test says. A laboratory test is only as reliable as its analytical chemistry. Failing to bypass assay crosstalk with an MMA functional marker guarantees that severe neurodegeneration will proceed completely undetected. Once the diagnosis is clear, doctors must avoid the most severe medical error associated with this entire system. If a clinician sees the huge, dysfunctional red blood cells of macrocytic anemia on a chart, they might simply prescribe high-dose folic acid to fix the blood, without definitively excluding a B12 deficiency first. That massive dose of folate feeds the DNA synthesis pathway directly. It completely bypasses the stalled B12 engine, forcing the bone marrow to wake up and rapidly pump out normal-sized red blood cells again. The anemia disappears. The patient's blood work suddenly looks fantastic. But the folate did absolutely nothing to restart the second mitochondrial mutase engine. Toxic MMA continues to pile up. The faulty fats continue to destroy the myelin. The spinal cord silently decays in the background, while the doctor and the patient falsely believe the disease has been cured. Treating macrocytic anemia with folic acid alone is exactly like dealing with a glaring check engine light by cutting the wire, while the engine itself is actively on fire. It guarantees permanent, irreversible paralysis. To put out the fire and bypass the broken gastrointestinal gauntlet entirely, clinicians administer intensive, daily, deep intramuscular injections of hydroxycobalamin, manually restocking the hepatic vault. Preventing this catastrophic collapse requires adhering to 5 core architectural pillars. B12 is an ancient organometallic node, reliant on a fragile, multi-organ relay. It powers both a rapid DNA engine and a critical myelin insulation engine. Standard total serum tests are routinely compromised by inactive carriers and assay crosstalk. Mass spectrometry of plasma MMA is the only definitive marker of cellular starvation. And finally, single-agent folate masks terminal nerve decay. We are complex biological machines, yet we remain absolutely beholden to the microscopic rules of ancient bacterial chemistry. Understanding these mechanisms and practicing rigorous laboratory stewardship is the ultimate guardian of our structural architecture.